Hello students, welcome to readmedprep Academy channel. Today in Human Health and Diseases Part 5, we are going to talk about the cells of the immune system, antigens and antibodies, and antigen antibody interactions. Let us move on to the cells of the immune system. The immune system is composed of many interdependent cells that protect the body from microbial infections and the growth of tumor cells. All these cells are derived from pluripotent hematopoietic stem cells. Each stem cell has the capacity to produce RBCs, WBCs and platelets. Here in this table you can see the cellular composition of the adult human blood, the cell type, the number of cells per microliter and approximate percentage. The red blood cells of the RBCs are around 4200,000 to 6500,000. The white blood cells, A granulocytes, the lymphocytes are around 1500 to 4000 per microliter. It is about 20 to 30%. The monocytes are 200 to 950. They are about 2 to 7%. Granulocytes, the neutrophils, around 2000 to 7000 per microliter, about 50 to 70%. Basophils 50 to 100, less than 1%. Eosinophils 40 to 500, about 2 to 5%. Platelets 150,000 to 500,000 per microliter. You can see in this image the different types of blood cells. The monocytes, the large cell in the blood. The lymphocytes, which has a large nucleus with the rim of cytoplasm. The neutrophils, which has a multi-lobed nucleus. The eosinophils, the bilobed nucleus. Basophil, having the large stained dark granules. Macrophages, which are irregular in shape, which are the main elements for phagocytosis. The erythrocytes, which do not have the nucleus or the RBCs. And the platelets, which are small components of the blood cells. The only cells capable of specifically recognizing and producing an immune response are the lymphocytes. The other types of white blood cells play an important role in non-specific immune response, antigen presentation and cytokine production. Now lymphocytes, about 20 to 30% of the white blood cells are lymphocytes. They have a large nucleus filling most of the cells surrounded by a little cytoplasm. The two main types of lymphocytes are B and T lymphocytes. Both these are produced in the bone marrow. B lymphocytes, otherwise called the B cells, stay in the bone marrow until they are mature. They circulate around the body. Some remain in the blood while others accumulate in the lymph nodes and spleen. They have receptors on the surface. When receptors on a B cell bind with an antigen, the B cell becomes activated and divides rapidly to produce plasma cells. The plasma cells produce different types of antibodies. Some B cells do not produce antibodies but become memory cells. These cells are responsible for secondary immune response. In the image below on the right side, you can see the B lymphocytes having large number of different types of receptors on the surface of the cell. Activation of B cells, you can see when any antigen in the form of a virus or a bacteria enters into the body, it is received by the B cell and gets attached to it, and the B cell activation occurs and it gets transformed into a lymphoblast. This lymphoblast helps in the production of a series of plasma cells which helps in the formation of antibodies, and sometimes this B cell becomes a memory B cell. T lymphocytes, they leave the bone marrow and mature in the thymus gland. Once mature, T cells also accumulate in the same areas of the body as B cells. Lymphocytes also have a receptor proteins on their cell surface. T lymphocytes do not produce antibodies. They recognize the antigen presenting cells and destroy them. The two important types of T cells are helper T cells and killer T cells. Helper T cells release a chemical substance called cytokine, which activates the B cells. The killer T cells move around the body and destroy the cells which are damaged or infected. Here in this image you can see when an antigen enters into the body, it combines with the antigen presenting cell and gets attached to the T lymphocytes which gets transformed into a T helper cell or CD4 plus cell. And these T helper cells helps in the proliferation of the B cells and the plasma cells and the production of antibodies. And these T helper cells sometimes stimulates the macrophages to favor or increase the phagocytotic activity. Some T helper cells get transformed into the killer T cells. You can see the expansion below. APC means antigen presenting cell. MHC2 is called major histocompatibility complex of class 2. Apart from these cells, neutrophils and monocytes destroy the foreign cells by phagocytosis. Monocytes, when they mature into large cells, they are called macrophages, which perform the activity of phagocytosis on any foreign organism. Here in this image, you can see how the cells of the immune system are formed. In the bone marrow, there is a pluripotent stem cell which gets differentiated into a myeloid progenitor and a lymphoid progenitor. The myeloid progenitor helps in the formation of the erythroid progenitor, which results in the formation of the series of cells resulting in the formation of erythrocytes. The myeloid progenitor helps in the formation of basophils, platelets and eosinophils. The pluripotent stem cell gets transformed into a granulocyte monocyte progenitor and multiplies rapidly to produce large number of neutrophils or monocytes, which helps in the transformation into macrophages and the dendritic cells. The lymphoid progenitor that is formed from the pluripotent stem cell forms the B cell series and the T cell series. Dendritic cells are called so because it's covered with the long thin membrane extensions that resemble the dendrites of the nerve cells. These cells present the antigen to the T helper cells. 4 types of dendritic cells are known: Langerhans cells, interstitial cells, myeloid cells and the lymphoid cells. Here in this image you can see the formation of dendritic cells and their origin. The hematopoietic stem cell gets transformed into a common myeloid progenitor, which forms the Langerhans cell, interstitial dendritic cell, or sometimes it helps in the production of monocytes, which gets transformed into myeloid dendritic cell. The hematopoietic stem cell forms the common lymphoid progenitor, which results in the formation of lymphoid dendritic cell. Now let us move on to the antigens and antibodies. What are antigens? The term antigen AG is used in 2 senses. The first to describe a molecule which generates an immune response, and the second a molecule which reacts with the antibodies. In general, antigens are large complex molecular substances that can induce a detectable immune response. Thus, an antigen is a substance that is specific to an antibody or a T cell receptor and is often used as a synonym for immunogen. An immunogen is a substance capable of initiating an immune response. Here you can see a pathogen having a large number of surface antigens which are different in specificity and get attached to the antibodies. What are haptens? Haptens are substances that are non-immunogenic but can react with the products of a specific immune response. Substances that can enhance the immune response to an antigen are called adjuvants. Here in this image you can see that hapten molecules themselves do not elicit an immune response. But when they are combined with a carrier molecule, they form a hapten carrier conjugate, and this induces an immune response in the body. What is an epitope? Epitope is an antigenic determinant and is the active part of the antigen. What is paratope? It is the antigen binding site and is a part of an antibody which recognizes and binds to an antigen. Here in this image you can see the epitope, which is the active part of the antigen which combines with the paratope. The paratope is a part of the antibody which combines with the epitope. The histocompatibility antigens are called cell surface antigens that induce an immune response leading to rejection of an allograft. You can see in the image a donor tissue being transplanted into the recipient tissue. The recipient T cell directly recognizes the donor tissue and causes acute graft rejection. Or sometimes the T cell receptors gets attached with the recipient cells and the donor antigen presenting cell is formed by the dendritic cell, and this exposes itself to the recipient antigen presenting cell, and this indirect recognition results in the chronic graft rejection. What is antigenicity? It is a property of a substance or an antigen that allows it to react with the products of the specific immune response. What are types of antigens? On the basis of origin, antigens are classified into exogenous antigens and endogenous antigens. The antigens which enter the host from outside into the body in the form of microorganisms, pollen dust, drugs or pollutants are called exogenous antigens. The antigens which are formed within the individual are called endogenous antigens. The best examples are blood group antigens. Here you can see the differences between the exogenous antigens and the endogenous antigens. Antigens that enter the body of the organism from outside are called exogenous antigens. Antigens produced from within the cell as a part of the normal cell metabolism or when the cell is infected by bacteria or viruses is called endogenous antigen. Antigen that can enter the body through ingestion, inhalation or injection are called exogenous antigens, whereas endogenous antigens do not enter the body. They are byproducts of the cellular metabolism. Example of exogenous antigens, pathogens like bacteria, viruses, etc. or allergens like the pollen and the toxic food. Example of endogenous antigens, byproducts of regular cell metabolism or molecular components of pathogens inside the infected cells. Exogenous antigens are non-self antigens, whereas endogenous antigens are either self or non-self. Exogenous antigens occur in the body fluids and the extracellular space, whereas the endogenous antigens are presented on the cell membrane. Exogenous antigens are presented along with the major histocompatibility class of antigens 2 molecules, whereas endogenous antigens are presented along with the MHC class 1 molecules. What are antibodies? Antibodies are immunoglobulins, which are protein molecules synthesized on the exposure of antigen that can combine specifically with the antigen. Whenever pathogens enter our body, the B lymphocytes produce an army of proteins called antibodies to fight with them through the plasma cells. Thus, they are secreted in response to an antigen by the effect of B cells called plasma cells. The antibodies are classified into 5 major categories based on their physiological and biological properties and biochemical properties. They are IgG gamma, IgM mu, IgA alpha, IgD delta and IgE epsilon. On the right side you can see the antibody being combining with the antigen. In the 1950s, experiments by Porter and Edelman revealed the basic structure of the immunoglobulin. What is the structure of antibody? An antibody molecule is a Y-shaped structure that comprises of 4 polypeptide chains. 2 identical light chains called the L chain of molecular weight 25,000 Daltons, approximately having 214 amino acids, and 2 identical heavy chains called H chains of molecular weight 50,000 Daltons, approximately having 450 amino acids. On the right side you can see the Y-shaped structure of the antibody having 2 white chains close together and the 2 light chains away from the heavy chain on either side. The polypeptide chains are linked together by disulfide bonds. 1 light chain is attached to another heavy chain and 2 heavy chains are attached to each other to form a Y-shaped structure. Hence, an antibody is represented by H2L2. The heavy chains have a flexible hinge region at their approximate middles. Each chain, light and heavy, has 2 terminals. They are C terminal called the carboxy terminal and the amino or N terminal. Each chain, light and heavy, has 2 regions. They have variable region called the V region at one end and much larger constant region called the C region at the other end. Antibodies responding to different antigens have very different V regions, but their C regions are the same in all antibodies. Here you can see the structure of the immunoglobulin. It has 2 heavy chains linked together by the disulfide bonds. Each heavy chain is linked on either side by the light chain through the disulfide bonds. And the light chain and the heavy chain have a constant region which is fixed in all antibodies and a variable region which can vary in different antibodies, and it is the part where the antigen binds. In each arm of the monomer antibody, the V regions of the heavy and the light chains combines to form an antigen binding site, shaped to fit a specific antigenic determinant. Consequently, each antibody monomer has two such antigen binding regions. The C regions that form the stem of the antibody monomer determine the antibody class and serve common functions in all antibodies. Here you can see the image where it has two heavy chains, and on either side of the heavy chains is the light chain. The two heavy chains are linked by the disulfide bonds, and the heavy chain and the light chain is linked by the disulfide bond. And the constant region is present, which is the same in all antibodies in the light and the heavy chain. Whereas the heavy chain and the light chain have variable regions to form the antigen binding sites. In this image you can see the antigen bound in between the variable regions of the light and the heavy chain. The functions of immunoglobulins are agglutination, precipitation, opsonization, neutralization, etc. Now let us move on to the antigen antibody interactions. The reaction between an antigen and antibody is the basis for humoral immunity or antibody mediated immunity. The reaction between the antigen and the antibody occurs in three stages. During the first stage, the reaction involves the formation of antigen antibody complex. The next stage leads to visible events like precipitation, agglutination, etc. The final stage includes the destruction of the antigen or its neutralization. Here you can see in this image, an antigen is a substance or molecule when introduced into the body triggers the production of an antibody by the immune system, which will then kill or neutralize the antigen that is recognized as a foreign and potentially harmful invader. These invaders can be molecules such as pollen or cells or bacteria or viruses. In the image below you can see the immune cell binds with the antigen, and the activated immune cell makes more number of antibodies to the plasma cell, and the antibody and the antigen binds together. That is the first stage. And the antigen and the antibody are easily recognized by the macrophages and they are devoured. And the third stage, the macrophage destroys the antigen by phagocytosis, by lysis of the antigens. The binding force of antigen and antibody is due to three factors. They are closeness between antigen and antibody, non-covalent bonds or intermolecular forces, and affinity of the antibody. When antigen and antibody are closely fitted, the strength of the binding is great. When they are apart, the binding strength is low. The bonds that hold the antigen to the antibody combination site are all non-covalent in nature. These include hydrogen bonds, electrostatic bonds, van der Waals forces, and hydrophobic bonds. Antibody affinity is the strength of the reaction between a single antigenic determinant and a single combining site on the antibody. In the image you can see below on the right side the antigen on one side and the antibody on the other side, and these are bound by the hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals interactions. What are the chief application of the antigen antibody reactions? To determine the blood groups for transfusion, to study the serological ascertainment of exposure to the infectious agents, to develop immunoassays for the quantification of various substances, to detect the presence or absence of protein in the serum, and to determine the characteristics of certain immunodeficiency diseases. Different types of antigen and antibody reactions. Precipitation reaction. The reaction between soluble antigen and the antibody leads to a visible insoluble precipitate formation, which is called precipitin reaction. Antibodies that bring about precipitate formation on reacting with the antigens are called precipitins. In the image on the right side you can see on either side the soluble antigens and the soluble antibodies. When they are combined together, they form a precipitation zone where a precipitate is formed which is insoluble. Agglutination reaction. Whenever a particulate antigen reacts or interacts with its antibody, it would result in clumping or agglutination of the particulate antigen, which is called agglutination reaction. The antibody involved in bringing about agglutination reaction is called agglutinin. In this image you can see the antigen combining with the antibody forming the clumping of cells. Opsonization or enhanced attachment is the process by which a pathogen is marked of ingestion and destruction by a phagocyte. Opsonization involves the binding of an opsonin, that is antibody, to a receptor on the pathogen cell membrane. After opsonin binds to the membrane, phagocytes are attracted to the pathogen. Here in this image you can see the complement recruitment by antibodies. The bacterial cell, when it attaches to the antibody, there is triggering of the complement component formation. The antigen antibody complexes, they activate the complement system, the classical pathway, and triggering its antibacterial activity. The phagocytic cells grab the antibodies that are bound to the surface of the foreign substances for efficient phagocytosis. So opsonization is the process in which the pathogens are coated with a substance called opsonin, marking the pathogen out for destruction by the immune system. This results in much more efficient phagocytosis. Neutralization reactions. These are reactions of the antigen antibody that involve the elimination of the harmful effects of the bacterial exotoxins or a virus by specific antibodies. Thus, neutralizing substances, that is antibodies, are known as antitoxins. These neutralizing substances, that is antibodies, are known as antitoxins. This specific antibody is produced by a host cell in response to a bacterial exotoxin or corresponding toxoid, inactivated toxin. In the image on the right side, you can see a virus infected cell. A virus combines with the antibody and the virus is inactivated. Antibodies bind and inactivate the viruses and the toxins. These antibodies are called neutralizing antibodies. To sum up the entire events of the antigen and antibody interactions, neutralization you can see where a foreign virus or a bacterium resulting in the neutralization of the exotoxin or the toxoid. Agglutination, formation of a clumping because of the particulate antigen combining with the antibody. Precipitation, formation of a precipitate due to the soluble antigen, the soluble antibody combining, resulting in the formation of an insoluble precipitate. The complement system or opsonization is triggered because of the foreign body entering into the cell and the antigen combining with the antibody. The complement system coats the antigen and helps in easy phagocytosis. So the neutralization, the agglutination and precipitation reaction causes phagocytosis by macrophages. The complement system also favors increase in the phagocytosis, causes release of complement substances and chemical mediators which causes inflammation, and it also causes cell lysis. So today in Human Health and Diseases Part 5, we discussed about the cells of the immune system, antigens and antibodies, antigen antibody interactions. So thank you. Kindly subscribe, like, share and comment the channel readmedprep Academy. Kindly register for UG and PG NEET type MCQs in our website www.readmedprepacademy.com. Our Facebook ID is readmedprep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is readmedprep Academy. Kindly WhatsApp the number given below to join readmedprep Academy for preparing for NEET exam. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.